Today's topic is about something called gene editing. Very simple. Uh, I'd like to just put a smaller thing on this. So fundamentally, you know the concept of editing. So when do you edit? When there is a mistake or you want to change. Any of these two quantities are really required. When you want any of these things, you will do editing work. So just like you're on your Microsoft Word, what you do, you cut it and then you rearrange it and then you paste it. So what you do is you cut and then paste the new thing. That is fundamentally what happens with respect to editing. So on a similar basis, the concept of gene editing. So what happens here is the concept is very simple. For example, if this is your cell, inside the cell you have nucleus. In the nucleus you will have sets of chromosomes. So these are all chromosomes. Now these chromosomes are the genetic material. So if there is any disease, for example, sickle cell anemia is there with you, which has been recently in news. So you can use this gene editing process to fundamentally clear that kind of disease. So what happens is here the chromosome is there. Now let's assume that the defective chromosome piece is something like this, and let's say this part of the coding is the reason why a particular disease happens. So what happens is there are two people who will come. They'll come here, they will slice this or they will cut it out, and along with them they bring the better part and attach it to it. That is what happens in gene editing in very simple words. The defective chromosome in that small part is cut. The body mechanism will realize, okay, there is a change in the chromosome, somewhere there is a problem. So obviously every body mechanism will have its own genetic material development mechanism or machinery in it. So once it realizes that something is wrong, the change has happened, this new structure will get added to here and a new chromosome which is undefective, not defective, will be formed. And slowly the body machinery will start accepting this format. This is called gene editing. So it has been recently in news because of wanting to utilize gene addition process to clear sickle cell anemia. So under that process, this news has been brought, therefore the concept of gene editing. So let's look into what is gene editing and all that. So under that, please look here, CRISPR, clustered regulatory interspaced short palindromic repeats. This is what you have to remember for your prelims. CRISPR, clustered regulatory interspaced short palindromic repeats. This concept utilizes the process of gene editing fundamentally. So how it is done? Please look at this one. What is it all about? It is a genome editing technology. It removes, adds or alters the sections of the DNA sequence. So in this majorly there are two parts. I told you there are two people who will come and remove and cut and paste. So these guys are those fellows. One is Cas9. Fundamentally this Cas9 is actually an enzyme. What is an enzyme? Enzyme is nothing but it is a protein. What is the function of this protein? It actually acts as a catalyst for faster, since it is a catalyst, it accelerates the chemical reactions and the sequencing within the body. So this is fundamentally an enzyme, it's a protein and it accelerates reaction. So that is a key molecule. So what it does is, it works as the scissors part, the cutter who cuts the defective gene. So he does that. But how does this fellow know, how does Cas9 know which part of the chromosome to cut? There should be a guide There should be a teacher or a guru who should tell, okay, here is where you have to cut. There should be an identifier. That identifier molecule is gRNA. So fundamentally, this is a second key molecule, and what this fellow does is, it finds and binds. It finds, he comes, cuts it, binds a new one, and a new chromosome is created. And that is usually undefective. So this is what happens in this technology. The next part, please look here. What happens? DNA editing, Cas9 first key molecule, it acts as the scissors. This, the GNA, this works as find and bind. So the guide is this RNA, ribonucleic acid, and cuts across both the strands of the DNA. Cell recognizes the DNA of the damaged part, and the DNA repair mechanism is used to introduce the changes. This is what happens in, how the process happens is this. So editing work. So these two are very useful for your prelims. So Cas9, and what is Cas9? Fundamentally, I told you, it's an enzyme. Enzyme means it's a protein which accelerates chemical reactions. Very simple. Next, what is the good part of it? It is faster and cheaper, high accuracy, and it is viable compared to genetically modified crops. And there can be a huge development in health. This can be utilized even in agriculture practices. And example, Japan is using for tomato currently. So these are the paths. This you can use it mains ke liye as an example. Fundamentally, you can say the gene editing can be used to bring a lot of change in agriculture as well as human health services. You can just use it for that. And this can be used for prelims apart from the molecules of that. So good part. What is a defective part? The biological concern is, once the chromosome changes, for some time, let's say everything is fine. The problem with chromosomal changes, there are a lot of mutations which happen. Mutation means changes. Let's say if the changes are for good, excellent. But what if the changes suddenly mutate in such a way that it becomes bad? The problem is, the mutations with the current generation will be transgressed to the next generation. That means from the father, it goes to the kid, and from the kid, it goes to the grandkid, and it becomes inheritance of defective pieces. And that is a problem. So see, this process is not 100% accurate as of now. Therefore, the problem is that. So mutations will have side effects. So transgression to the next generation, and this is one thing. Usually currently, though it is cheaper, even the cheap cost is very costlier for the poor. So if gene modification happens and gene editing services are brought into the market, it is always captured maximum by the rich. And again, there will be a huge difference for gene editing process between the rich and the poor. Therefore, that gap is called gene gap. That's the point. And moving on, for India, what is the situation? It is still an infant stage. It is growing steadily. Problem is of funding and investment. So which committee does that? Genetic Engineering Appraisal Committee. It comes under the Ministry of Environment, Forest and Climate Change, and final technical body that certifies a genetically modified product as safe for commercial release. So what you have to remember for your prelims is this: Genetic Engineering Appraisal Committee, and it comes under Ministry of Environment, Forest and Climate Change. This is what today's topic is all about. So that is called CRISPR. Very important. Thank you.